So what I would like to do in the next 30 minutes or so, and I promise to be even shorter, we are late, is to tell you about the initial efforts to implement a personalized oncology program here in Italy within the framework of a network of research hospitals, so-called ACC, Alleanza contro il Cancro. So the ACC network includes 21 cancer research hospitals, which are called IRCs, so recognized as of strategic relevance by the Ministry of Health. The network itself is coordinated by the Ministry of Health and has a number of affiliated local hospitals. So the potential of this network, both in terms of clinical and research is remarkable, I guess. Clinically, we're talking about 90,000 new cancer patients every year, 70,000 patients in clinical trials, and more than 5,000 active clinical trials. And a remarkable research performance, as you see by the indicators shown there. So the network collaborates actively with the Patients Association and with the Ministry of Health for NIHS regulations, which are really important, I guess, for the implementation of precision oncology nationwide. I guess, I mean, this program started only less than 2 years ago. The kickoff meeting was June 2016, so it's very, very young. And I guess the only advantage of being late, if not too late, is that of building up on other countries' experience and starting from failures rather than successes. So what I would like to do is just to review which are the challenges of precision oncology, which have been judged as such by this community of scientists, and which are at the basis of the strategy of this program. Then tell you a little bit about the first national screening that we have just started on lung cancer, and a very, very brief mention to the ongoing activities which represent the major challenge of our program. I think there is little doubt, as we heard before, on the potential, on the disruptive potential of precision medicine, and particularly precision oncology, mainly in terms of improving public health. And I also guess that in the long term, this will be sustainable. So in other terms, it is perceived that the precision oncology might be economically viable despite skepticism on the short-term implementation. And this is built up on very successful stories which have really changed the natural history of some specific tumor types. I reported in this slide, which is the only slide in this context, just the first and the last example of successful curative treatments in oncology using targeting agent. The first example is a rare disease. It's a subtype of myeloid leukemias, the acute promyelocytic leukemia, which is now the first example of a otherwise fatal disease which can be cured without chemotherapy using a combination of targeting agents, in this case retinoic acid and arsenate. And we had the privilege to contribute significantly to the build-up of this story by identifying the genetic lesion, the mechanism of targeting by both retinoic acid and arsenate, and also in the setting up of the clinical trials, which have shown the most recent in 2012, the curative effect of the combination of targeted drugs. So no chemo for this type of disease. And the last example, I don't have to comment because it has been done by previous speakers, is the immunotherapy with checkpoint inhibitors, and other success stories in the middle of these two examples. However, a number of challenges, of limits have been clearly emerged in the last 10 years. Some of them have been alluded by the previous speaker, and I will very briefly mention them. The first point is the number of eligible patients which actually access treatment with targeting agents. Not easy to have definitive figures on how many patients which can access, because are eligible, new drugs actually receive that treatment. Probably in Italy it's less than 20%. And this is due to many different reasons. First of all, because the omic approaches are not yet sufficiently standardized for clinical usage. Second, because the resources which are required are not easily sustainable in a routine clinical setting, both in terms of cost, time, and also human effort. And there are limited screening capabilities and also drug availability. All things which limit the access of patients which could benefit to actual benefit of available current treatment. So this sets one of the priorities of this program, which is to guarantee access of patients to screenings and available drugs at the national level. The second is the actual number of eligible patients. If you do exercise with in silico prescription tools, just challenging the 15,000 cancer genomes which are available, asking the question how many of those patients could have been treated with available target agents, just restricting the analysis to FDA-approved drugs within guidelines, it is less than 20%. There are, however, a lot of drugs in pipeline, so this percentage of patients, so low patient of patient that can benefit of the FDA-approved drugs goes up probably to 60–70% if we consider repurposing and the combination of drugs, which sets another goal of this project, which is that of guaranteeing access of the patients to the clinical trials. Third point has been mentioned, I go very fast, is the actual cure rate from these drugs, which is limited. Most of the available drugs are not curative. The issue is clearly that of resistance, which dominate over sensitivity. And the other issue is the pure value of the stratification markers that we have. More efficacy in predicting resistance rather than sensitivity to the available drugs. So there is an urgent need, in my opinion, to renew efforts in fundamental cancer research. The issue of tumor heterogeneity is a gigantic issue. It's at the basis of treatment failure, and we know very, very little about that. Then there is a technical problem, so-called technical, which is that the number of info that we need to rationalize this type of treatments is amazing in terms of number of biomarkers per patient, the number of potential options per biomarker, and putting together genomic information and knowledge is not easy. And we do need prescription tools. We do need, first of all, to collect and handle really big data, and then to allow the doctor not to have to study medicine, but to acquire information within the patient and decide for each single patient. And that needs some electronic tools. So the general goal based on that of this program, this newborn program, is that of use now all the information which is available to improve treatment for the patient population. And that's a goal which is that of doing whatever we can do now with the available drugs. The second is that of trying to continuously transfer new genomic discoveries into clinical practice, which is very difficult. And the third one is that of leading the transition from single marker genomic markers to omic scale diagnostics, take advantage of other omic approaches which are providing digital information on disease and sensitivity to drugs. So genomic, metabolomics, and so on. The short-term goal are that of promoting national programs of genomic screens and genomic basic clinical trial, disseminate genomic capabilities, mainly focusing on the creation of new professionals like clinical bioinformaticians, for example, which is one of the main goals of this program, and set up an infrastructure to handle data and to generate prescription tools. So the sequence strategy that we decide to adopt is purely based on money. So the idea is that if we want to reach as a target the Italian population, we need to sequence at low cost. So these are the figures which are already old, as I will mention at the end, of cost of sequencing when we made this decision, which is 1.5 years ago, not that far. So as you can see clearly, you cannot afford a genomic screen for the entire population with whole genome sequencing, so whole exome sequences, which is far more than 1,000 sample. And so we decided to go targeting selected fraction of the genome, so-called gene panels, whose cost, referring only to the labor and to the reagent cost, is below 500 euros. And actually, we decided to set 500 euros as cost per patient, not per sample, as the max limit to implement a nationwide approach, as you'll see. So basically, the sequence strategy was that of using NGS-based panels, very particular one, so home-designed, high sensitivity, very flexible to be able to modify them quickly based on new discoveries, and as I said, low cost. So having to decide where to focus, we decided to focus only on the actionable genome. We define the actionable genome through our actionability database, and which is basically putting together all available database with our own effort on literature mining, machine-assisted literature mining. We ended up with 166 genes whose mutations are linked to drugs. So basically 162 genes linked to 485 drugs. And that's what we defined as the actionable genome. 485 drugs include all the FDA-approved drugs for treatments in oncology, approved for other indication, and the clinical trials. We didn't include any drug in preclinical pipelines. So we worked a lot in this 1.5 years to create the infrastructure. So we have now a bioinformatician community, a wet lab community. So all the 21 centers have acquired NGS technology. 14 are ready to start and actually started to sequence. So the idea is not to bring the DNA where the sequence machine is, but to bring the machine where the patients are, and create a centralized infrastructure that we created within the Elixir node here in Italy. And I would like just to mention two of the efforts of this IT infrastructure, which is the prescription database, which is a database where we put together genomic information both from patients and the available public resources, which are linked to info, knowledge, and also to clinical data. That's the interface of this prescription database, which is a working in process, which allow the doctor to access all the available information with respect to the specific genomic markers that characterize that patient. I guess the graphics is not great, it can be improved, but the info are there. And also we worked on trying to elaborate tools to facilitate clinical trial designs. That's a tool, the Precision Trial Designer, which has been done in collaboration with Luca Mazzarella and Giuseppe Corigliano, which basically provides a number of parameters which are critical to the design of genomic-based clinical trials, using the information which is genomic information which is available on the web, particularly the TCGA, but not only, which allow to predict the average number of biomarkers per patient, the biomarker co-occurrence, the probability of drug allocation conflicts, and the number of patients needed to sequence. I guess we need to facilitate the design, not only in terms of innovative design and of one adaptive platform, but also in terms of prediction of the segmentation of the population in terms to maximize info from the clinical trial itself. This tool has been validated using the Shiva trial data in collaboration with Christoph. That was so nice to share all the data from the clinical trial you heard about it. Okay, so the first clinical trial that we started, as I mentioned, is this national screening of lung cancer. So that's the organization of the network in many disease-oriented groups. And the idea is, the goal, the screening started in November 2017, so a few months ago. And the goal is that of sequencing 1,000 lung cancer patients in these 21 centers. It's a prospective trial, clearly, in order to quickly assign patients to the best treatment. And the main purpose is that of identifying in the network which are the obstacles for the precision medicine approach in terms of turnaround time, sample preparation, drug availability, and a number, I don't go over, of intermediate goals which aim at standardizing and unifying the sequencing producer pipeline, the analysis pipeline, and finally the access to the information database. So this is the lung gene panel that we are using, which we have generated in this last 1.5 years. So as you can see, it's a relatively small gene panel. The genomic space which is occupied is less than 0.7 megabases, and it costs less than 500 euros. It contains the 164 actionable genes I was referring before, and 70 translocation fusions. Plus, we also put a limited number, 30, of drivers, not actionable, as derived by the available driver identification pipelines. So we validated using standard cell lines, which carry Cas9 engineered mutations to validate the accuracy of the panel. So I don't go over it. I would just mention key points, like we optimize protocol for 20 nanogram of DNA, which is critical, especially for lung cancer, where the material is never too much. And it is clearly a DNA-RNA workflow, because the effort in identifying the diffusions, and it has been optimized on low-quality paraffin material, both DNA and RNA. We set up a robust bioinformatic pipeline for calling SMV and for the RNA fusion. We paid particular attention to the CMV calling. We think that it's problematic in gene panels as compared to genome-wide or whole exome sequencing. And we developed the proprietary pipeline in collaboration with Maggi in Florence, which is part published and another one is submitted for publication right now. So this is the performance of this lung cancer panel, which is clearly the critical point. And as you can see, 97% of the patient this is challenged against the 500 lung cancer patient of the TCGA using the PT tool that I mentioned before. 97% of the patients have at least one actionable mutation. I'm not referring here to drivers, only to this 160 actionable genes. And each patient has approximately 4.5 average mutations. So patients show more than one actionable mutations, posing again the question of the treatment choice. So this is ongoing, and the goal is to reach the 1,000 patients within this year. Meanwhile, we have set up a so-called second-generation gene panel, which we have validated technically, as you will see. And the reason with which the approach from the tumor-specific panel, the lung cancer, is because in 1 year the price went down again in terms of sequencing cost, and we can increase the size of the explored genome by the same price. And in fact, we have now this new panel, which is around 3 megabases, which is now a pan-tumor. It includes, of course, the few available markers for immunotherapy stratification. We have enlarged significantly the number of translocations that we can detect. But the most important thing, in my opinion, is that this includes also germline actionable genes. So genes whose mutations, actually, whose variants predict cancer risk. Again, we have defined the germline actionable genome, which we ended up with 120 cancer predisposing genes, which are genes for which there are clinical evidence of association with increased risk. And the numbers are pretty high in terms of frequency of these variants, not only the ovarian that we all know, but also the breast, which if you go down to, for example, triple negative, it goes up to 20%, the frequency of germline actionability, and so on for the other tumors. And this allows to propose a slight switch of the approach to the patient based on the fact that we can, when the patient comes into our hospital, simultaneously map both stratification markers as somatic mutations and germline variants, same moment, which allow a sort of stratification for treatment and bottom-up build-up of a registry of cancer risk variants, and of course tracking then family members. That's the detection power of this new gene panel, which is 315 actionable mutation per patient, very, very variable depending on the tumor type, as you can see here. I don't have time to go into details, but the detection power in different tumor types differ significantly. And the validation, as I was mentioning, is ongoing, and we changed the target approach, and we're now using hybridization instead of amplification, which allows a better recovery of background baseline genome sequences to identify mutational load. And it also includes a molecular barcode for high sensitivity assays. Okay, 3 slides to mention, to share with you, which are the ongoing activities. Again, and this is endless, I guess, we are changing our sequencing strategy. The reason being that the price went down even more. We can now, with the last Illumina machine, the NovaSeq, at least in our hands, do a whole exome sequence with less than 500. So there is no point in gene paneling. Clearly, switching from panels to whole genome sequencing, it is tough, not only for the cost, but because it imposes a different logistics, and it imposes a even stronger effort in terms of bioinformatics. So we decided to start 2 pilots. One is for RNA-seq analysis in breast cancer, with a specific goal of extracting the very well-known RNA prognostic signatures from RNA-seq, so to compare them with gene panel analysis. And the other one is to do a pilot on AMLs for whole exome sequencing. Again, I guess switching from panels to whole exome is, if it is not a problem of money, it becomes a problem of logistics and manpower in terms of analysis. And so we decided to start small pilots. Liquid biopsy, we have a strong plan together with Bardelli and Turin, that of developing new generation high sensitivity approaches. We have set up, adapting a published protocol, duplex type of sequencing that, as you can see, allow to go very deep in terms of low VAF identification. For example, these are the results of the sensitivity of these methods in tumors. And you see that you go down to 0.001%, and right now NGS doesn't go, for theoretical reasons, below 5%. And now we are adapting that to circulating DNA. And the last, we are discussing a lot on the opportunity to start real-world clinical trials, as opposed to investigational pilot clinical trials. And this is based on data that probably you know better than I do, which are very, very worrisome, in my opinion, with respect to the value of the new drugs in real-world health. And in fact, there are papers on both sides of the ocean that show that the effects of the new drugs after the entry of the market in real-world health is nothing or marginal, with percent which are 49 and 51%. And this clearly may be due to different reasons. I mean, the registration studies are not aimed at identifying optimal treatment, so you cannot expect that the real world they work. They are not aimed at defining optimal treatments. And also we frequently give these drugs to patients based on indication which are not that of the registration, think to the off-label or the compassionate use. Whatever is the reason, we risk to kill the value of these drugs if they are not used in a proper context after market entry. So we are now designing a real-world clinical trial, pretty similar to the TAPUR clinical trial, which has been started in the United States, which aims at assessing safety and efficacy of only the FDA-approved anti-cancer drugs, exclusively in the context of genomic-based clinical trials. We're talking about 22 FDA-approved drugs coupled with 48 genomic abnormalities, so very defined, clearly including also repurposing of those drugs with respect to the approved guidelines, and placed into a real-world health context. So any patient which has a molecular target, molecular biomarker being identified, being eligible for this type of clinical trial. And the main point is that of collecting data to share with the scientific community and also with the big pharma. And I thank you for your attention.